Okay. So in systemic circulation, low levels of oxygen in a particular area of tissue. So we're talking about kind of just the local control now. And again, if there's low oxygen, that means you got to send more blood into that region. So that means you need to increase the flow, which means you dilate the vessels. And so we have local vasodilation. This is increase of mean arterial blood pressure. That's not something that is being controlled by some little local area of tissue. That's kind of the big picture. That's being controlled by the overall system. Veins, everybody got this, tend to have thinner tunica media. They don't need as much muscle to hold their diameter because the pressure in them is so much lower. Yeah, and actually everybody got this one too. The, where is it? About 3 liters a day leaks out of the capillary beds. And what happens to that, all that fluid that leaks out into the interstitial space? Yeah, it gets picked up by those little lymphatic capillaries, kind of filtered through the lymph nodes, and ultimately, where does it end up? It just gets dumped back into venous circulation. It goes to the main lymphatic vessels, the right lymphatic duct and the thoracic duct, all go into the vena cava, basically near the brachiocephalic vein. Describe at least 2 ways. This I just haven't created them all. Most people got this. How are ways that blood keeps moving in the veins, even though it starts out really low, like 20 millimeters of mercury, it goes down from there. How do we make sure the flow keeps moving?
Being massaged by skeleton.
Yeah, so there's that, and that only works because what else is in place in the veins? The valves, right? So if you were just squishing them without the valves, the blood would just be squishing. But because you have those one-way valves, when you squish, the blood can only move back towards the heart. So this one-way valves, the massaging of the skeletal muscle, we also talked about the pressure changes in the thoracic cavity due to breathing, also help drive the flow of blood back to the heart. The specialized capillaries in the bone marrow, spleen, and liver, those were the sinusoidal capillaries. Those were those really kind of big kind of places with gaps in the walls where cells can actually reach in and help clean out the blood and things like that. They're more intense. The fenestrated capillaries are leaky, but they don't have big gaps for cells to reach in to the blood and stuff like that. So that is that.
I have a question.
Sure.
Sorry, how does the pressure in the thoracic cavity push the blood through the veins?
Because the vena cava and as well as the atria that are receiving the blood are in the thoracic cavity. So they are in that space that is going higher and lower pressure. So that's what's happening. Yeah, as the kind of atria get kind of pulled out by the whole expansion of the thoracic cavity, that creates kind of a low pressure that helps push the blood, pull the blood in from the other places. Get that out of the way. So we've been talking about vessels. I realize the one last thing we need to add in to kind of complete our discussion of vessels are some of the kind of pathologies of vessels. Talk about aneurysms. What is an aneurysm? So it's a weakening of the vessel wall. So if the vessel doesn't have the ability to kind of maintain its diameter under the blood pressure inside, you get this kind of ballooning out of the wall. If this is kind of a cross section. That's an aneurysm. Just in and of itself, it can cause problems because it's going to be putting pressure on surrounding structures around it. But much more concerning is, the fact that it might burst, in which case you're going to have a major hemorrhaging going on. If it happens, if your aorta, an aortic aneurysm is not that uncommon. If that bursts within a 30 seconds, you're going to be dead because all the blood is going to just spew out of your aorta into your thoracic cavity. If you have an aneurysm in one of the major cerebral arteries that bursts, you're also going to be dead pretty quick. The other ones are not going to be as fatal, but still pretty bad. In general, if they find aneurysms, they try, if it's small, they might just monitor it over time to make sure it's not getting worse. If it looks particularly concerning, there are different ways to try to stabilize them. Putting things around them. There's things where you can add little metal coils even to try to solidify the area in there. There's a variety of different interventions, but typically, if it looks like it's worrisome, it has a potential to burst, they're going to try to stabilize it. Hardening of the arteries. This is when you start getting these atherosclerotic plaques. So plaque is these lipid inclusions. As well as kind of a proliferation of the smooth muscle. It's happening inside the wall. It's in the tunica media. So it is impinging on the diameter, increasing resistance to flow, right? If this is happening in your coronary arteries, it's gonna make it harder and harder to get sufficient perfusion of your cardiac muscle. This is happening in your carotid arteries. It's gonna make it harder and harder to get the blood up to your brain. So this can be worrisome. Things to note, people often think about like the fat actually being inside the vessel itself. You're eating all that fat, it's clogging up your arteries, which is not really what happens. The lumen is still lined with simple squamous epithelium. This thing is happening inside the wall of the vessel. And it's kind of indirectly. There are correlations of eating high diets, high in saturated fats and having more incidence of this, but it's not because the fats stick into your vessels. It's because through a more indirect process, there are these inflammatory processes that get triggered in the metabolizing of the saturated fats that ultimately result in the formation of these plaques. So it's more indirect. Your diet can affect the incidence of these, but it's more through this inflammatory process that gets triggered. And it's partly also, you know, genetics. You know, the same people eating the same diet are going to have more or less of this kind of stuff. What else about them? Less likely to happen with like unsaturated fats. So anyway, so those are two of the issues that happen with vessels that we should be aware of. Okay, the last thing we need to talk about with the cardiovascular system, which will also be what we're doing in lab, is blood. So we're going to talk about blood a little bit. Right. All right, blood. So obviously it's what's flowing through your vessels, and it's what's helping transport all the stuff we've been talking about, those nutrients, the oxygen, the wastes, and everything. There are two main components: plasma and formed elements, which are mainly the cells. Now, if you remember from anatomy, blood is actually considered a connective tissue. It's a fluid connective tissue, where the plasma is the extracellular matrix, the kind of stuff around the cells, and then the formed elements are the cells that are kind of widely scattered within the extracellular matrix, within the plasma. Typically, the ratio of plasma to formed elements is somewhere around like maybe 55% plasma to like 45% formed elements. But it varies, and it's one of the things you'll find out exactly what it is in your own blood later this morning. Basically, if you take the blood and put it in a little capillary tube, you're just gonna take your blood, you're gonna be sticking your fingers later on this morning and bleeding and letting your blood fill up a little capillary tube, which is gonna be a mix of cells and fluid. And if we wanna just separate them, pull all the cells down to one side and leave the fluid up on the other part, what do? Centrifuge. So we're gonna have a little centrifuge. We'll centrifuge them. And after you centrifuge them, all the cells will be back on the bottom. There'll be the plasma on the top. And you can just figure out if this is the total X. This is what we call this total height. This is the X, the height of your cells. So for the total height, 100, basically the percent of your overall blood that is cells, we call that hematocrit. So for, on average, hematocrit is about 45%. About 45% of your overall blood, total blood volume is made out of cells. It varies from person to person. It tends to be higher in guys compared to women. So, when we do our lab today, we'll actually, there's a couple of guys. So yeah, we'll be able to look at Ethan and Paolo on average compared to everybody else. See if that goes up. So hematocrit, again, it's part of our lab today. Let's talk a bit more about plasma. Plasma is mainly water. It's like, I'd say like 92% water. Then there's a bunch of dissolved stuff in it, like nutrients and waste. And 1% is going to be dissolved nutrients, like, you know, their blood is carrying glucose around and whatever else. It's carrying urea around, you know, so waste. It's carrying like hormones, whatever else. So all that stuff that's dissolved in there is taking up a little more of the volume, right? So, but it, you know, it doesn't add that much volume. If you have a cup of coffee and you add in a spoonful of sugar and stir it up, you don't see the amount that's hiding your cup change all that much. But you can have all that stuff dissolved as just solute in the plasma, in the water of the plasma. And that leaves us with 7%. And these are going to be all of these plasma proteins. These plasma proteins are doing all sorts of things. You've already met some of them in the renin-angiotensin mechanism, right? We talked about angiotensinogen just floating around in the bloodstream, ready to go. That's a plasma protein. When we talk about blood clotting in a few moments, all of those enzymes and stuff and different like fibrinogens and prothrombins and all the stuff that are involved with blood clotting, they're floating around just as proteins. When we get to the immune system, we're going to see all the complement proteins and stuff. There's all sorts of proteins that are involved in your immune response. They're doing all sorts of jobs. The main one, maybe 60% of this is albumin. Yeah, albumin is just a protein. One of the things that this does is it helps preserve the osmotic balance between the plasma and the cells. So these, obviously, you saw what happens if you put red blood cells in water. It's not going to end well for the cells. So you need to make sure that you have enough stuff dissolved in the plasma so that the cells are going to be happy. So this albumin plays an important role in, you know, equalizing the osmotic balance with the cell, bringing our osmolarity of the plasma up to 0.3. You know, also limes, cherries, like non-polar things. So there's also like alpha globulins, beta globulins. They also, right, if you have something in your blood that is non-polar that you want to carry, and there's a lot of things that are non-polar that you want to transport in the blood. There are steroid hormones are non-polar. We talked about them being derived from lipids, from cholesterol. There are fat-soluble vitamins that you want to carry around in your blood, but they're not going to just dissolve in water. So anything that's non-polar that you want to move through the blood has to be complexed with something that is polar. So as a big unit, it can actually flow through the bloodstream. Like all the sex hormones, like testosterone, estrogen, they're non-polar, they're steroids. They don't go into solution in water. So they have to bind to carrier proteins. And then the hormone on the protein can then move through the bloodstream. And particularly with those kind of hormones, they usually talk about bound hormone versus free hormone. How much of the hormone is just bound on a carrier and being transported around versus free and actually able to bind to receptors and cause some effect. So again, same thing with fat soluble vitamins. They're going to need something to carry them around. When we get to just in general transporting cholesterol around and stuff, we'll get even more dramatic with these high and low density lipoproteins, these big complexes of fat and protein that can then travel through the bloodstream. But just being aware, if you want to move a non-polar thing through the bloodstream, you need to get a little more involved with binding it with some protein so that the overall thing will dissolve, even if the non-polar thing in and of itself is not going to dissolve. So in general, these are all made in the liver. We are going to be talking about the liver later on when we get to the digestive system. But the liver does so many different things. The liver does so many things. It's hard to kind of pigeonhole it in any system. One of the things the liver does is makes all these plasma proteins. There's one other plasma protein that we'll actually get to meet a little later today when we talk about blood typing, which are the gamma globulins. So the only kind of plasma protein not made by the liver are basically the antibodies, which are made by lymphocytes. So gamma globulins, also called immunoglobulins, also called antibodies. And these are made by special lymphocytes. And we're going to talk about the antibodies in much more detail when we get to the immune system, but we do need to introduce them today because they are necessary to understand blood typing and the importance of blood typing and how we actually do it and all that. So these are going to be something kind of fundamentally different from all the other plasma proteins in that they're made by lymphocytes, these white blood cells, and are part of your immune system. Oh, actually, some of these other things are part of your immune system, like the complement, but these ones are specific. Again, we'll talk more about antibodies in a little bit, actually, when we start the lab. So this is plasma. Plasma got a variety of stuff in it. The formed elements, let's talk about the formed elements now, the blood cells and platelets. So far and away, the most common of the formed elements of the blood cells are the red blood cells, the Rbcs. Also called erythrocytes. It just means red cells. So all the blood cells are made by red bone marrow. This is, you should know this word, hematopoiesis. This is the formation of blood. Hematopoietic tissue is the tissue in your body that can make blood cells. Hematopoiesis, this happens in red marrow. Red marrow is within in a baby, it's everywhere because babies have to make tons of red blood cells and stuff. But in an adult, it's mainly in your hip bones and your vertebrae, and like the proximal ends of your humerus and your femur, and your skull bones. But that's kind of it. The rest of your bones as an adult are just filled with fat particles. Red marrow, and we have these cells that can make the stem cells for blood or hemocytoblast cells, which become like everything. The ones that are going to become red blood cells go through this process where they start making lots and lots and lots and lots of hemoglobin. And then ultimately, actually, once they are done with all of the protein synthesis and stuff, they eject most of their organelles. They get rid of their nucleus. They don't have a mitochondria. They don't have mitochondria. So a red blood cell ends up looking puckered because there's no nucleus, there's no mitochondria. But there are somewhere around 250 million molecules of hemoglobin in that. Because there is no nucleus, once they are out in the bloodstream doing their thing, they've lost the ability to kind of maintain and repair themselves. They no longer have the blueprints and all of that. So these things only last a few months, around maybe 3 or 4 months. That's why they're constantly getting recycled. We talked about the spleen being a red blood cell graveyard, or in the sinusoidal capillaries of the liver. You're constantly recycling the red blood cells, and new ones are constantly getting made. Let's talk a little more about hemoglobin. So hemoglobin has quaternary structure, which means what? The protein.
More than one tertiary protein.
It has more than one tertiary subunit. So we have these 4 globin subunits. Which are made out of amino acids like a regular protein. And then there are these iron-containing heme groups. So that is one hemoglobin. It has these 4 protein subunits, and it also has these 4 iron-containing parts, these heme groups. Each heme group has the ability to bind 1 molecule of oxygen. What does that mean in terms of how many oxygen molecules can 1 red blood cell carry?
Oh, one red blood cell.
1 billion.
1 billion, right? 1 billion molecules, right? We have 250 million molecules of hemoglobin, and each one can do 4 oxygens. That's 1 billion molecules of oxygen on 1 red blood cell. So these things are optimized to be transporting oxygen around. They also are going to be carrying a sizable portion of our carbon dioxide through the bloodstream. Maybe 30% of the carbon dioxide transported in your blood is bound to hemoglobin as well, but it's bound to the globin subunits. So carbon dioxide will, so hemoglobin isn't just about transporting oxygen. It's also about transporting not all of your carbon dioxide, but about like a third of the carbon dioxide. And there's a interaction between the carbon dioxide bound to the hemoglobin and the affinity of the heme groups for the oxygen. So hemoglobin. We're going to talk about this in more detail when we get to the respiratory system next week. But the basic idea here is the more carbon dioxide that is around, probably the more oxygen the tissue needs to. So like the more carbon dioxide that ends up in the tissues, the less hemoglobin is actually holding onto the oxygen, and the more likely it is to let go of oxygen into the tissue to let it to deliver it into the tissue. We'll talk about this in much more detail next week. The whole oxygen hemoglobin dissociation curve, that higher levels of CO2 as well as other things that would be related to like higher metabolic activity in the tissue will reduce hemoglobin's affinity for oxygen and therefore release more oxygen into the tissue. So that should make some intuitive sense. There's more metabolic activity in the tissue. We want to like, let's not hold on to the oxygen on the red blood cell. Let's let it go where it's needed. Remember, affinity is how tightly the two things are holding on to each other. All right, RBCs. So iron, and we'll talk about different kinds of anemia. Anemia is when you have reduced ability to deliver oxygen to your tissue. Different kinds of anemia. We'll describe them while we're here. You don't have enough iron in your diet to make the hemoglobin, you're going to have trouble making the hemoglobin. Your body is really, what do you call it? Like it holds on to iron. You don't like get rid of it. We're going to talk. In fact, when we go into the recycling of the red blood cells in the liver and in the spleen, part of that recycling, there's some of it that gets thrown out of the body. Part of the byproduct of recycled heme groups and stuff is bilirubin, which is what's making your poop brown. It becomes part of your bile. But all the iron is held in the body and then reused. It's part of the reason why it's not that uncommon for like little kids to get poisoned if they find iron supplements and gobble them down. Right? There's a lot of things, if you eat too much of them, you'll just pee out the excess. But iron, if you eat too many iron supplements, you'll actually get poisoned because your body isn't going to release it. Yeah, so if you have like little iron supplements around, those should be looked away. So iron deficient anemia. What are other ways you can get anemia?
Like sickle cell.
Yeah, sickle cell anemia. Sickle cell, this is when there is literally one base pair is wrong in the code, the genetic code to make hemoglobin molecule. And what happens is it makes the hemoglobin kind of pull on itself in this weird way, particularly under low oxygen conditions. And the whole red blood cell, instead of looking like a little flying saucer, ends up looking more like a canoe. And then once that's the why it's called sickle cell. And then those all kind of clog up in the capillaries and aren't flowing properly. So you're not delivering blood to where it needs to go. So sickle cell is a genetic problem with, again, one base pair off. If you have like one hit of it, it's not fatal. And in fact, it seems to be adaptive in environments where there's a lot of malaria. But if you have two hits of that bad gene, it's not going to be okay. What else can cause anemia? Not having enough red blood cells around, that can happen just from really bad bleeding. You have hemorrhagic anemia. This is not how you spell it, but there's most words in my life I am like rock solid with. This one just always spins around in my head. Anyway, 2 R's.
Yeah.
So just from bleeding. There are, aside from iron deficiency anemia, you can also have anemia from lack of vitamin B12. It's called pernicious anemia. Vitamin B12 is necessary for the proper formation of your red blood cells. So without it, you're not getting enough red blood cells and you've got problems. You know, so these are all types of things that can cause a lack of ability to deliver adequate oxygen to your tissues. So anemia, red blood cells. All right, and we'll come back to these guys in other when we talk again, when we get into the respiratory system, talking about moving carbon dioxide and oxygen around and all of that.
Good question.
Uh-huh.
So the other ones had kind of specific names: sickle cell, hemorrhagic deficiency. Is that just kind of like a garden variety anemia, or does it have its own special name?
I think that's what it's called.
Iron deficiency.
Iron deficiency anemia. It's just if you're not getting enough iron in your diet, that can lead to anemia. In lab, we'll also talk about the external markers on the red blood cells, the things that cause the type A, type B, Rh markers and all that. That'll be another important thing about red blood cells, but not for right now. Okay, so RBCs. These are like maybe 99% of the fourth elements. Are almost all of your blood cells. Then there are the white blood cells. Wbcs, leukocytes. These are fundamentally different. These are part of your immune response. We are going to talk about these in much more detail when we get to the immune system. So these are different. They have nuclei. They are not confined to the closed circulatory system. Yeah, there's a lot of white blood cells floating around in your closed circulatory system, but if they are flowing around and all of a sudden they get this chemical message that they are needed out there in the tissue, They grab onto the wall and they squeeze out through the wall and just go where they're needed. And a lot of them are never even in the bloodstream. A lot of them are just like, your Langerhans cells just patrolling in the layers of your skin, and Kupffer cells just hanging out in your liver, and all these other lymphocytes just moving around through the tissue spaces and hopping into the lymphatics and going from one lymph node to another. So there's a lot of white blood cells that do not live in the circulatory system at all. But that being said, there are white blood cells in your circulatory system and zipping around and listening for these little chemical messages that tell them that they are then needed someplace.
Which are the ones that are just patrolling in the skin?
Those are the epidermal macrophages, also called Langerhans cells. Again, we are going to talk about these in much more detail. There's going to be the lymphocytes, which are key players in your adaptive immunity. Maybe I should just spend a lot of time talking about B lymphocytes, T lymphocytes, plasma cells, memory cells, all sorts of cells that are involved with specific immunity. The ones making the antibodies are lymphocytes. The things multiplying and swelling up your glands when you've got an infection are lymphocytes. So we'll spend more time talking about these dudes.
And that's just a type of white blood cell?
It's a type of white blood cell. Yeah, so these are all subtypes of white blood cells. Monocytes, which turn into macrophages, which are these big eaters. They're the things that they're like big amoebas moving around and grabbing things and eating them, and then actually presenting them to lymphocytes and saying, look what I just caught, and let's set up some immune response against them. There's neutrophils, which are kind of more like the infantry of your white blood cells. Like they go where there's some infection and they start eating up all the bacteria, and then they're like all that white goo when you've got pus and an infection. It's like the dead and dying neutrophils that have come in and done their job, but now they're done. There's basophils, actually for like inflammation. They release histamine. We'll talk about inflammation in a lot more detail. Basophils, they're similar to mast cells in your connective tissue. And when they release the histamine, it initiates inflammation. They are eosinophils. They're more for like parasites, worms and stuff. These tend to be more effective against parasitic and multicellular things in your body, as opposed to these things which are eating like a bacteria. Again, we're not going to get so much into these. We'll spend a lot of time talking about macrophages and lymphocytes. And I should mention, this is literally in the last week or two, stuff has been published about red blood cells, that red blood cells are not completely one-trick ponies. You know, basically, when people think about these, they think about them transporting oxygen, maybe some CO2, and you know, that's it. It now looks like they actually can absorb bits of genetic material from invading things, which then they ultimately transmit to white blood cells when they get recycled, and then they actually can, so they are actually involved in presenting stuff to the immune system in ways people never knew about. At least that's what it's looking like from some of the latest work on them. But like literally, it's like reading that in the last weeks. It's nobody's ever like, at least I've never heard of that before. So just as a heads up, the stories always change. You know, we get deeper. The big picture, I don't think we're ever going to say red blood cells aren't really important for delivering oxygen. But, you know, the full story is probably more complicated than that. Neutrophils as well. Neutrophils people just usually thought mainly as first phagocytes. And then there are phagocytes that also have this kind of toxic cloud they can release to kill things. And now it looks like they also can like set up these nets that can trap things and keep them from moving around. So like neutrophils are way more like complicated than the initial stories when we first thought, oh, they're like phagocytes. So the immune system in general is still a major work in progress in trying to understand what the heck's going on with it, how it works. So white blood cells, we will come back and talk about them, but not right now because we're going to move on to our final topic in the blood, which is hemostasis, which is stopping bleeding. Well, I did mention this before, but it's worth probably adding into this moment now. Red blood cell production obviously is going to be modulated by the body's status on oxygen delivery. Right? If oxygen delivery is kind of low, that's going to cause the kidneys, actually. The kidneys release this stuff called erythropoietin, and that's going to upregulate red blood cell production. We have more of these little oxygen carrier molecules around to carry the more limited oxygen. Like when you go up to Tahoe, you know, the partial pressure of oxygen is less. We'll talk about that in much more detail later. But basically, that's going to cause erythropoietin to be released. By kidneys. Right, so if we have a harder time loading oxygen into the blood because of lower partial pressures, we're going to have more RBCs to carry the oxygen around. All right. Hemostasis, right? Obviously, something like the aorta, which is basically like a garden hose, is not gonna rip unless there's, you know, like kind of a horrible car accident or something. But as you get into the smaller and smaller vessels, they get really delicate, and down to the capillaries where they're like basically like wet tissue paper. So the vessels are constantly ripping just through moving around or bumping into things. So you need to have a system in place that is constantly going to stop that from happening. And that is going to be with the one last formed element, which are the platelets. So I talked about RBCs, white blood cells. There are also going to be platelets. They're not actually cells. They are made by these things, there's these big cells called megakaryocytes. Karyocytes in the marrow. And they basically reach into the blood vessel, which is the blood vessel. pinch off little sacs of membrane filled with a variety of different kinds of granules. So the platelets are these little bags of stuff. They're not actually cells themselves. They're little, like when you look at a slide of blood under the microscope, you'll see the red blood cells, you'll see the white blood cells with their nuclei, and you'll see those little things that look like the shimmerts around. Those are the platelets. They're just like these little goopers in there. But they are critical in hemostasis. So let's talk about their role in hemostasis. So normally, if blood is going through a vessel and the vessel is intact, what's the only thing the blood is ever going to see? Looking around the walls of the vessel. I'm inside a blood vessel. What's the roof? What's the floor? What are the walls?
Intima?
What's that?
Tunica
It's
intima.
the intima, the endothelium, the smooth, simple squamous epithelium. So as long as that is all I see, then it's cool. Then nothing is there's no evidence that there's been any breach. If there is a breach, then all of a sudden I'm going to be exposed to connective tissue. What's the main protein that is the fiber in all your connective tissue, you know, from anatomy? What's that?
Fibrinogen?
No, fibrinogen is something very different. That's a plasma protein involved in clotting. What's the main protein that makes up the fiber? Like leather. What's leather made out of?
Collagen.
Collagen. It's collagen. So collagen is the main protein that makes up the fibers and most of your connective tissue. It's what cartilage is made out of. It's the organic part of your bone. It's the tough part of your skin that keeps your skin from ripping. So this is initiated. Platelets are exposed to collagen. So if the platelets see collagen, they know something's wrong, right? It would be like if you're in the room and you see 2x4s and conduits and stuff. It's like, okay, something happened that ripped a hole in the wall, right? You should not see the collagen if you're in a intact blood vessel. All you should see is the endothelium. This is going to initiate a whole cascade of events, which we will kind of briefly summarize here. So the first thing that happens when the platelets see the collagen is they can actually release. I talked about how they have all sorts of different little granules. We'll talk about the different kinds of things these granules do as they get released into the body. They actually release serotonin. Which causes vascular spasms. So serotonin in this context, like we've been talking about serotonin as a neurotransmitter in the brain that's getting mimicked by LSD and magic mushrooms and stuff. This is where people first saw serotonin. That's where it kind of gets its name, actually, is causing constriction of vessels. Why is causing vascular constriction going to be a helpful thing in this case?
Reduce blood flow.
Yeah, less blood flow, right? There's a rip in the wall. Okay, let's shut down the flow into this region so we don't just bleed out here. So this is going to decrease the amount of flow into the area. Platelets also release other things, ADP, etc. Track other platelets. Yeah, spiky and sticky. They basically form what's called a platelet plug. So maybe like here was our vessel. And then we got a rip in the wall. Get the vascular spasm where the vessel actually kind of constricts to reduce the blood flow into the region. Now the platelets start this thing where they send out these messages to attract other platelets, and they all get spiky and sticky, and they make this platelet plug. It's kind of like sandbagging. There's a leak and you put a bunch of sandbags there to try to keep the blood from flowing out. So that's so this is kind of in the short term. Let's reduce flow and sandbag the hole. Now we're going to start some more intense kind of repair stuff. We're going to start the whole blood clotting cascade. They call this clot formation. This is going to be lots of steps. I should tell you exactly how many steps. We have here. Yeah, so there's ultimately there's over 30 different substances involved, where the platelets start releasing stuff, which then does this kind of whole cascade of events using different plasma proteins. I'll just give you the last two stages. So I'll say like, over 30 steps. Last stages are, there is a plasma protein called prothrombin, which is normally just a plasma protein floating around, and things called pro whatever are in this version that's not active yet. So finally, the thrombin, and then thrombin is actually a catalyst that converts another plasma protein called fibrinogen. Which is soluble. Into fibrin, which is insoluble. So fibrinogen is a soluble plasma protein. It's just normally in your plasma flowing around in your blood. But when thrombin is created as a result of this big cascade of events, it turns the fibrinogen into fibrin, which is these sticky insoluble threads, which are going to create the clot. Kind of like a Band-Aid there. So this is, it's a lot like a Band-Aid. So the platelet plug was more like kind of sandbagging, but now we've actually got these sticky threads kind of sealing off the rip. Fibrin clot is going to be, that's when you think about, when we talk later about kind of clots, that's what this is, it's that fibrin. In our lab, we're going to actually see fibrin with our own eyes. You're going to bleed into another little tube and break it after a minute, 2 minutes, 3 minutes. At some point, you're going to see, oh my God, there's that weird thready thing that's showing up in my blood. That's clot formation. That's showing the fibrin, which was an insoluble fibrinogen, which is just going to make look like water. But when fibrin is created, all of a sudden you've got this physical thready thing showing up in the blood. Okay. Obviously, the next step is going to be like we're in the moment, it's all cool, but we want to fix it. So the next part is going to be what we call clot retraction and repair. Part of this is going to be initiated by another thing that the platelets release called platelet-derived growth factor. So this PDGF basically helps regrow the muscle and epithelial tissues here and fix it. Clot retraction means there's contractile proteins that kind of squeeze things together, squeeze all the kind of fluid out of this big mess here. So clot retraction and repair is like going to pull everything together and actually fix the actual tissue of the vessel. So the vessel is complete again. Once the vessel wall is actually complete and healed, then the very last stage is going to be to dissolve the clot. We don't need that big vibrant clot there anymore. So part 5 is going to be dissolve the clot. That is the job of yet another enzyme that is created from one of these plasma proteins. There's a plasma protein called plasminogen, which becomes plasmin, which dissolves the clot. So plasminogen, which is just already there, at this last stage gets converted into plasmin. Plasmin is an enzyme that dissolves fibrin. And the fancy word is fibrinolysis, because you're lysing fibrin. Right, so then the fibrin clot is gone. And now we're back to where we started. So are there any questions about these different steps? Yeah?
How does plasminogen know what converts it to plasmin? Like when it's
Right, so
obviously you
it's
can't
got to
start
be
doing that.
some, I'm not exactly sure. I'm sure it's something that gets triggered once the repair is done. There's got to be some signal that goes out that converts the plasminogen to plasmin. But I don't know exactly the details there. I can say though, the first ever commercially viable product from a biotech company was from Genentech. It was TPA. TPA, which is basically tissue plasminogen activator. What do you think this is going to be used for? People would probably, if you worked in a ER or something, you might have actually shot up somebody with this stuff.
I remember that it had something to do with strokes.
So it has a lot to do with strokes. With stroke is often when there is some thrombus that breaks free. So a thrombus is any kind of clot that becomes kind of errant. That could happen just because. It can happen because the blood isn't flowing well, it's pooling because of AFib or something. But if you have a clot that forms just because, if it breaks free and it's an embolus is if it's floating free, and as it goes along, maybe it's fine in the bigger thing, but then it gets to a smaller branch and now it's blocking the flow. And now the blood can't move on, and if that's leading to your brain, you've got a stroke, right? So if you have some errant clot, some little thrombus that broke free and is blocking the arteries leading to your brain, unless you can deal with this in the near future, you're going to have permanent damage to your brain. So if I think you have to do it within like an hour or two. If somebody presents with a stroke, you can shoot them up with this stuff, turns on plasminogen into plasmin, which can dissolve the clot. And it's we call it like a clot buster. It'll dissolve the fibrin and restore the blood flow. But obviously it has to happen. You can't repair the damage downstream if the tissue's been deprived of oxygen. It can only get rid of the clot and restore blood flow. So you have to do it fast. What else? Yeah, so thrombus, they're not so good. If people are at more of a danger of having these form, again, like if you have atrial fibrillation or different conditions where people are just more likely to get these, then you are going to want to have things that inhibit clot formation. Things that inhibit clot formation, aspirin, right? There's people who take little baby aspirin every day to prevent stroke. It's because it's minimizing the chance that a thrombus is going to form. We're going to do this in our lab today. We'll have some of you take aspirin as we begin the lab, and as we do the time for clot formation, we're going to be able to compare the people who took aspirin compared to the people who did not, and see how much that affects the ability of the blood to clot. Other things, heparin, which is warfarin. Often people call these like blood thinners. I'm going to put it in quotes. But they don't actually thin your blood. What they do is they reduce its chance of forming clots. So if somebody is in danger of like these thrombuses, these clots, they'll be on these blood thinners. The dosage is really tricky. You have to really titrate the dosage. If you don't have enough, then you're not going to get the protective benefit you're looking for. But what happens if you have too much? Yeah, it's like people will start just getting all bruised up just from trying to put on their socks and stuff. Because every time you're doing and pushing on your skin, you're getting little rips here and there that are instantly sealing up. But if you have too high a dose of these things that inhibit clot formation, these blood thinners, then you're going to start bruising all over the place and having the little tears not heal up like they should. There's some, I forget the name of it. There's the newest one of these drugs, I think, is a little less tricky with the dosing. But in general, the dosing of these things, you have to really monitor because there's kind of a sweet spot where they're doing their job, but not actually causing trouble. There's much trouble with it. Heparin, we're going to have heparin actually inside the little tubes that we use for the hematocrit, right? Because we're going to put our blood in a little tube and spin it down in the centrifuge to separate the cells from the plasma. But the moment, we don't, if the blood is all turned into, kind of fibrin goo, then the cells are not going to be able to spin down. So you need to make sure we have heparin in the tube, and that's going to inhibit the clot formation. So we can actually get the hematocrit. You know, in the tubes where we're going to be assessing the time for clot formation, obviously those have to be non-heparinized because otherwise you're not going to ever clot. What else? Clot formation. So anyway, hemostasis, obviously important, happening all the time, can cause problems. Think about this. Oh, so one last thing I'll mention before we kind of tie up the whole cardiovascular system. When I was talking about local autoregulation of blood flow. We talked about the blood's going into some region of your body. Low O2 is going to like increase vasodilation, increase the flow. What if they're chronically low oxygen? You know, the body's doing its little things to, you know, adjust the flow, but it's chronically not enough oxygen in the region that you're trying to deliver oxygen to. What can the body do? Long term, get angiogenesis. Angio just means vessel, genesis, creation. You actually increase the blood vessels into the region, so you have more perfusion today. So there are these little genetic programs you can turn on to grow new blood vessels and increase the blood vessels into the region. So that's another thing that the body can do.